computer, yes. Okay, hi everyone. Dr. Dominique Lessard here, Science Communication Director at KIF1A.org. I'm here today with Dr. Jayne Aiken, a postdoctoral researcher in the Holzbaur Lab at the University of Pennsylvania. Dr. Aiken has recently authored a research simplified resource for us entitled A Brief History of KIF1A, From Gene Discovery to Disease Mechanisms. And in this resource, Dr. Aiken chronologically walks us through the discoveries of kinesin motors, including KIF1A, then through some of the major findings that shape our understanding of KIF1A as we know it today, and lastly discusses some of the implications of certain KIF1A mutations. So Dr. Aiken, thank you very much for joining me today to discuss your Research Simplified article and help answer questions from our community. I think a great place to start is for you to tell us a little bit about your research background and perhaps how you got into the field of KIF1A research.
Absolutely. Thank you so much for inviting me here and for inviting me to provide this article. It was a wonderful experience for me to get to share this to a more general community, which always puts things into a different perspective for us scientists as well. So my research background starts with microtubules. As a graduate student, I studied tubulinopathy disorders, which are caused by rare mutations to tubulin genes. And the tubulins are what form microtubules. And that's what KIF1A walks along. So I was already really immersed in the neuronal microtubule cytoskeleton and trafficking, and felt like there were so many questions left open in this field about how microtubules and also the things that regulate them, like how they contribute to neurodevelopment, neuronal function, and importantly, disease. So when I was looking for a postdoc position, I had reached out to Erica Holzbaur, whose lab does fabulous work in this field, and she told me all about this great news story that a previous postdoc in her lab had just finished up, Pedro. He had looked at KIF1A. He actually did a research simplified on this research for the CAM community, and showed that microtubule regulation and KIF1A, like how it walks along those microtubules, that's important for synaptic function. And so I was really excited about this project and felt there were so many questions left to investigate. And so I really jumped headfirst into KIF1A research and have just been blown away by entering this community and seeing both the researchers as well as the families, all of the wonderful support for the research out there.
Well, we're so thankful to have you as a part of our community. And I love that answer because it really highlights the fact that there are so many different avenues in which people enter KIF1A research. There's not just one road. You can approach it from so many different perspectives. But thinking a little bit about your perspective in the lab, you know, when you're in the Holzbaur Lab and you're working on your projects, how do you study KIF1A? What questions are you trying to answer, or maybe what level of investigation are you pursuing in the Holzbaur Lab?
So I really go about my research using a two-pronged approach, which I think is really important. On one side of it, I go very specific into molecular mechanism. And to do that, the cell is a very complicated space. There's a whole lot going on, a lot of proteins, a lot of moving parts. And so to kind of get around that and to answer more simplified questions, we use in vitro approaches, which means that we take things out of the cell and can put them together in very simplified ways and look, well, okay, I see KIF1A, it's walking on a microtubule, I add something, how does it react? So that's one avenue of research that I've been actually able to get into the lab to do a lot recently. And then the other approach is using cells. So I'm really excited to be working with human stem cells that I can differentiate into neurons. I think this is a wonderful approach to use, especially in light of canned research, because you know, historically we've used a lot of model organisms, so using different animals to study these questions, but being able to look at KIF1A function within a human cell is going to be really informative. And so I can use these human neurons, manipulate KIF1A or other proteins, look at things such as synaptic function, cargo trafficking, all these different questions that we're interested in, and see in a human cell how it reacts.
Fantastic, thank you. I want to divert for a second to the content discussed in your Research Simplified article, particularly about this historical perspective of KIF1A discovery. So it mentioned kinesin motors originally discovered in the 80s, 85, then jump forward about a decade and we start to see the emergence of KIF1A in the 90s. And then quite a bit of time goes by, around 2011, where we start to see reports of KIF1A mutations on a clinical level in the literature. So we sort of have this gap in, we identified this KIF1A protein, but then this dramatic amount of time before we identified KIF1A mutation. Why wasn't a patient with a KIF1A mutation discovered before 2011? Why do you think that this gap exists?
I really love this question. I think it gives us the perfect opportunity to discuss kind of like the origin of sequencing technologies, because it's really fascinating. So actually the first paper that I mentioned that came out, there's actually a pair of them that came out in 1985 identifying kinesins. The DNA sequence is actually not published at that time. It's not until 1989 that the first kinesin sequence is published. And that's because historically it was really labor intensive to sequence a gene. It required a lot of work, and this technology was just emerging. And really it was throughout the 90s, beginning of the 2000s, culminating in 2003 with the publication of the Human Genome Project, that we really had this baseline code to compare things to. So basically the Human Genome Project was able to provide all of the sequence that is encoded in our cells, which is an amazing feat. It took over a decade. And then since then, both the technology and then having this kind of like roadmap to compare to has really enabled genetic sequencing to become much more prevalent. So with the advances in the technology, which made the process much more labor intensive, and importantly, brought the cost down a lot. We've been able to sequence people and patients, both for developmental things, sequence tumors, all the different kind of things, which help specialized medicine. For the example, so even though now we have this amazing tool in DNA sequencing, it's still really challenging to identify disease-causing mutations. Because if you think about it, then we take KIF1A for an example. In 2011, there was a paper published where they looked at a family and they had sequenced the entire exome. So the exome is all the parts of the genome that encode proteins. And then they had to go through, so that's a lot of code right there. And then they had to go through and look at all of this code to find mutations that were correlated to disease symptoms, because it gets a little bit more complicated. So even like within people, I have a number of mutations that are, aren't affecting different function of my proteins. They're called SNPs. This is kind of getting technical, but it's really hard to differentiate something that is normal and probably not doing anything negative versus something that is really correlated with disease. And so you have a lot of code, you have to go find these things, but then once you are confident, okay, we think that this gene and these mutations within this gene are disease causing, then you can go through, and if a patient presents with similar symptoms as the previous person, you can go through and test just a subset of genes and look for mutations within those. So it is quite complicated, and that's, I think, what led to this big gap between, okay, gene discovery and like association of the gene with disease. And then, yes, since 2011, so, just about a decade, what we've been able to discover about the patients, the diversity of symptoms and clinical manifestations, as well as function of KIF1A has really taken off.
Thank you. That's such a good description about the complexity of genetic sequencing and the history of it. And it also brings up a really good point that we see a lot with new techniques in scientific fields, which is that, just because a new technique has evolved doesn't mean it's necessarily the most accessible. And so that is another leap, whether that's, affordability for researchers or affordability for patients, always finding ways to identify those barriers and find ways that we can overcome them. I next want to talk about a concept that I think is hard to wrap your mind around if you're not necessarily a kinesin molecular biophysicist, if you don't think about kinesin motors in a mechanical way.
Yeah.
And it's this concept of dimerization. We've said it before that, two kyphone A proteins have to walk together on the microtubule, and that when we actually are showing these videos or cartoons of a kyphone A protein, it's actually two of them. So can you go over just this concept of dimerization a little bit more, and if you have any thoughts about what the implications of dimerization may be on certain therapeutic strategies?
Absolutely. So at a very basic level, as Dominique just showed, dimerization basically describes when two proteins are linked together. And there's a whole lot of chemistry behind what's bringing them together, how long they're going to stay associated with each other. We're not going to get into that, but basically, if you look at my hand and imagine that it is a kinesin dimer. So my palm here is basically the part that's linking them together, and then my two fingers are the motor domains, right? And so and then my arm here is the microtubule. So as a kinesin motor, and that's another confusing part. We say a kinesin motor, but it's really two kinesin motors that you need. But as this functional unit is walking, at any given time, as it's walking along, one of these little motor domain feet remain associated with the microtubule. And so you can imagine that if you only had one of these proteins, which we refer to as a monomer, every time it unbound, it could just float away, right? It's not like, us walking. I mean, you can hop because you've got gravity pulling you back down. It doesn't work like that in a cell at these very, very small scales. So if it unbinds and it doesn't have another foot that's already anchored in place, it's just going to float away. And therefore the cargo, my arm, whatever it was trying to transport, isn't going to get to where it needs to go. And so this, it's a really interesting thing for kinesin, because if you look at the literature, and we describe it as a controversy because one group would come out, and when it was identified, they were sure this is a monomeric protein. And then another paper comes out, well, KIF1A can't walk on microtubules unless it's a dimer. And then another paper came out, well, maybe it's a dimer sometimes, but a monomer other times. And, and it's just this like kind of rebuttal back and forth using different research approaches, different systems to kind of get a more complete idea of how it exists. And now the kind of overarching model, and this, has kind of progressed through, the 2010s. This is all very recent, actually, where now we believe that, yes, it does float around as a monomer in a cell. And those two dimers, it doesn't dimerize itself until it binds a cargo. And then once it's got its cargo together, then it can walk along the microtubule. And that's really important, actually. And kinesins, there's different strategies that make sure that these motors aren't just walking without their cargoes. Because you can imagine, if you have an empty train, the train can get there, but you didn't have any of the stuff that was supposed to be on the train, and that's not good either. And so the more that we know about the regulation of these motors and the different cargoes they need and how they work together, the more we understand about its function in the cell, as well as what we can do to fix what's going wrong. And like these things are very challenging, and there's no one right answer for that. But the more complete idea of what it's supposed to be doing, the more we'll be able to help fix it when it goes wrong.
Absolutely. And I think that's a great plug too for where basic science fits into therapeutic discovery. It's this having as comprehensive of an understanding as possible of this system so that we know all of the avenues in which we can investigate some sort of therapeutic agent. Absolutely. You had mentioned in your everyone's favorite dance move, that, talking about KIF1A cargo. And while we know quite a few cargos of KIF1A, we also don't quite have a complete understanding of the complexity of cargos that KIF1A carries. I think it's, after being investigated for so many decades, it's surprising that this is still a question. Why don't we have a complete understanding of all these cargos? Why is this still being investigated, do you think?
That is also a fabulous question. And it also gets into some of these sort of technical weeds of the tools that we researchers have at our disposal to investigate things. And basically, it all comes down to, as a researcher, if I'm looking at a cell, I can't actually tell where any proteins are unless I do something specific to look for them. So in the case of KIF1A, in the very first paper talking about it, it listed synaptic proteins as cargos of KIF1A. And they came to that conclusion by using things called antibodies, which we've heard a lot about in the news, but it's like, this is another thing that's confusing about science, is we have all these words that kind of have like dual meanings. But in our case, at a simplified level, an antibody that recognizes KIF1A will allow a researcher to be able to visualize where in the cell it is. And so they could look at KIF1A, see, okay, it's here, 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 right? And then they also looked at synaptic proteins and like, oh, it's also here, here, here. So probably KIF1A and those synaptic proteins are moving together. More recently, with the discovery of fluorescent tag proteins, which were originally found in jellyfish that fluoresce, we can do that in a real-time sort of way. So we can take the part of the jellyfish that makes it fluorescent and tag it onto our protein of interest, such as KIF1A. And then also researchers found ways to make these different colors. And so I can use one color for KIF1A, see how it moves along the axon and dendrite, take another color, put it on either a synaptic protein or a different type of vesicle, and then see, oh, these two different colors are moving together. That suggests that probably this one is a cargo of KIF1A. But, and then there's also functional assays. So, okay, I think this cargo is being moved by KIF1A. As a scientist, I can be like, well, what happens if I get rid of KIF1A in that cell? So we call this either a knockdown or a knockout, where we're taking out the protein. So if this cargo was supposed to move, and it normally does when KIF1A is present, if I took out KIF1A, it's not going to where it needs to go. And that's a really good indication that we've found a cargo that is actually being trafficked by that specific motor. More recently, there have been some really cool, like, inducible motor domain, like linkage experiments. I'm not going to get into the details of those, but importantly, it still relies on us tagging or somehow identifying a cargo that we're interested in. So basically, there are a lot of proteins in a cell. There are a lot of potential cargos. And all of these experiments that I've described are not trivial to perform. And so as researchers, it would be really hard to just, kind of march down the list of candidates and being like, okay, is this cargo? Is this not a cargo? And so like what we've discovered is ones that we're most interested in. And so that's kind of where the limitation comes in and why we don't have a complete, fully, yes, these are all of the cargos that KIF1A is important for trafficking. And I think that because of that, and hopefully with, increases in our technologies and different pull down, all these different systematic methods that are kind of emerging, we might be able to find more. But historically, we've been limited by, we can only see what we're looking for. And so that's one of the reasons why we don't fully know all of the cargos, I think. But it does provide, like, there are quite a few that we do know that I listed in my presentation. And that gives us a really great place to start, because a lot of these are important for synaptic function. A lot of the clinical manifestations of KIF1A associated neurological disorder have to do with potentially synaptic dysfunction. And so I think that that's a lot to be going off of now while we're still kind of searching for additional cargoes.
That's a great point. And a point that I was thinking of as you were talking about it is, unless you have an extensive amount of experience looking at cells under microscopes, you may not realize that cells are basically see-through in a microscope. We see all these cartoons of pink cells and green cells with blue dots, but they're clear. And so how do you know what you're looking for when you're essentially looking at like a water snake? Yep. That's a functional unit. And so it's a good plug for when we don't have that visual information, how much we rely on the information that we do know about KIF1A, or thinking more globally, what information can we get from the clinical level, from the organismal level, from the chemical level, that can help us identify targets instead of sort of just picking a protein and saying, maybe this moves, maybe it doesn't, having a little bit more of an informed decision.
Absolutely.
Great. Well, I want to wrap up quickly just with a question about sort of what motivates you. You've been a resoundingly active member in our research network, and we're so thankful for all that you've done. We're excited for our relationship to continue. But we're curious to know, where do you get your inspiration? What is your motivation? What pushes you to keep working through those tough days where experiments are just not working? You're working late hours in the night. What motivates you to keep going forward?
Yeah, and unfortunately that happens more than you would like.
Yep.
I think that all of us in science at some level just are fueled by curiosity. A huge part of what I do is it's just fascinating to be looking at something, and that moment where you have this hypothesis, you think that this is going to happen, and you can be looking, it can be happening in real time in these cells that you're imaging, and being like, I think that I'm right, and that this is, and discovering something that no one in the world knows. And I'm here in my little dark microscope room, and I'm finding this out. That, I think, is a huge, huge part of the joy that I get from science. The other, which has really come about with my involvement with the canned community, is seeing it's really amazing for me as a scientist to have a link to the people that I could potentially be having an effect on. Like seeing the community, seeing patients that have canned, and how it affects their life, their family's life, and just how supportive and amazing this connection is that between the families and these researchers. I as a person studying tubulopathies, there was no resource like this. And so for me, it was very black and white paper. I'd read about patients written, by some MD or PhD out there, looking at brain size or whatever. And there was definitely a disconnect. And being a part of this KIF1A.org community has really changed that for me. And, getting sort of weekly updates on how patients are doing and how, our canned heroes are doing is really a huge motivation for me in studying KIF1A and trying to make those discoveries in my little microscope room about how it's working and potentially how we can help, fix these problems, with the neurons and the synapses. All these basic things that I'm studying, has the potential to make a big impact. And being able to see that, through this community is really impactful to me and to my research, and really helps drive me.
Thank you so much. And I would just want to say, that goes both ways. I'm biased. I think our community is amazing as well, but researchers play such an important role in empowering patients, and making them feel seen, and giving them a voice that is hard to give unless you're in a research space. So from all of us here at KIF1A.org, thank you again for all the work that you do. Thank you so much for this Research Simplified article and this interview, and we can't wait to see where your research takes you.
Wonderful. Thank you for inviting me.
Thank you.